Assalamu alaikum wa rahmatullahi wa barakatuh. Welcome, my dear students of grade first secondary. Today, inshallah, we'll continue our lesson, the cell theory and the cell discovery. We are talking today about the microscope, the discovery of the microscope, and the development of the microscope. As we see in the picture here, we have a light microscope. This light microscope, can you see here? It has many parts. Some parts are called mechanical parts and some parts are called light parts. From the mechanical parts here, we have the base. This is the base of the microscope on which the microscope stands and rests. This is a base. And also from the mechanical parts, the arm. The arm you can see, you can hold the microscope from the arm. And also from the mechanical parts, you have here the stage that you put the slide on it, the stage that you put the slide on it. And we have here a tube. This tube has two ends, two sides. In the first side, we have ocular lens or the eye lens. This is ocular lens or the eye lens is from the light parts. We can say it is a light part, not mechanical part. And we have another lens, which is objective lens. And this objective lens is not one lens only, but many lenses, maybe 3 or 4, because you need to use different strengths of magnification, different power of magnification. So you have to know that the different microorganisms are the different cells that you are studying, depend on the size of the cell, depend on the size of the organism that you study. You can use different objective lens. For example, one lens from the objective lens, it has power of magnification 10 times. One lens has one lens of the magnification, its magnification power about 20, maybe 40 times, maybe one magnification lens 50 times. Welcome, Omar Hussein. Welcome, Omar Hussein.
Welcome.
How are you? Fine?
Yes.
We are talking about the light microscope now, Ya Omar, which we will use to study the cell, and we will use to study the microorganisms. We are talking about the light microscope. In the beginning, the light microscope that you can find in the school. If you go to the school lab, you can find this light microscope. Also, you can buy one for yourself to use it at home for studying some drops of water. You can study some microorganisms. You can study some cells like your blood. Here, there is a drop of blood. As you can see here, there is a drop of blood. We use the microscope to study the components of the blood. For example, like this one. This is a drop of blood under microscope. And we are talking about the microscope. We are talking about the light microscope. It has many parts. As you see, it has it has many parts. These parts, some of them are called mechanical parts, and some parts are called light parts. Mechanical parts and the light parts. For example, for the mechanical parts, we have here the base. This is a mechanical part which is very important because it is the base of the microscope that you set the microscope on it. The microscope stand on it and rest on it. And also you have the arm. The arm, this arm, like the elbow, yeah, this arm, you hold the microscope from it, okay? And you have the big adjustment and the fine adjustment. You have two adjustments, big adjustment and fine adjustment. This one is called the big adjustment, and this one is called fine adjustment. Here we have big adjustment and fine adjustment. We have two adjustment buttons that you can rotate. You can rotate to change the position of the tube. This tube you can move it up and down. You can move it up and down to make the suitable distance to see clearly, more clearly. This tube is very important because the tube carry the two types of lenses. The two types of lenses here, this tube carry them. This tube here, as you see here, the tube has two lenses. Here in your eye, we look at the lens here in front of your eye. This is an ocular lens. The ocular lens found inside the top of the tube. Or you can say another name, can be called the eye lens. The eye lens or ocular lens. Use this to look at the microscope from it. The eye lens. But not only the eye lens, we have also another lens from the light parts. We have also the objective lens. Okay, the objective lens is placed nearer to the object that you study, or the cell that you study, or the microorganisms that you study. You put the slide, the microscope slide here on the stage, and you focus the light on it by the help of this mirror. This mirror reflects the light on the stage, and the slide here, you have a slide, a microscope slide made of glass. You put, for example, a drop of water or a drop of blood to study, to examine by the microscope. And the stage it has a hole in it because the light must pass through it. It has a hole in it because the light must pass through the stage and penetrate the slide. So the slide must be very thin. The tissue which you study, if you want to study, for example, the tissue of the membrane, مثلا, from your mouth, or a membrane in the plant, any plant, like مثلا, you can study the tissue of you can study the tissue of the onion, the membrane of the onion, the inner epidermis of the onion. You put the epidermis of the onion, for example, you have a slide, a microscope slide like this. You have a microscope slide like this made of glass, and you put on it the drop of blood. You can put on it anything that you will study here. You can put on it the microorganism, or the drop of water, or the drop of blood, or the tissue that you will study. So this is called a slide, microscope slide. This slide you place here on the stage. You place here on the stage. This is slide. You place here on the stage. In this place, you place here here. You place it here on the stage, and the light penetrates the slide because the slide is very thin. It has a very thin thickness, so the light can penetrate it, and now you can look at the eye lens or the ocular lens, and the image is formed by the two lenses: the objective lens, which is nearer to the object, and the eye lens, which is nearer to your eye. Type. We have many objective lenses. Why we have many objective lenses? One, for example, here, two, and three. Why we have many objective lenses? Of course, because you have different objects that you study or examine by the microscope. So you need different power of magnification. You need different power of magnification. For example, one lens, for example, here has strong power of magnification. Its power may be مثلا 100 times, 100x. So the magnification of this lens is 100x. And the magnification of the other lens, for example, this one, may be 40 only, 40x, 40 times. The magnification, 40x means 40 times. Okay? And this lens, for example, magnification is only 20x. 20x. So you have now to change the objective lens as you want to use its magnification. And the ocular lens is one lens only. The ocular lens is one lens only, so its magnification is constant. For example, it may be مثلا 10. Its magnification may be a 10x type. What is the total magnification of the microscope? The total magnification of the microscope will be the multiply of the power of the two lenses. We have ocular lens and the objective lens. So if we want to calculate, the magnification power, of a microscope, will be what? The magnification power will equal the magnification power of ocular lens and multiply the magnification power of objective lens. The power of a ocular lens multiply the power of what? The power of a objective lens. So you change the power as you want. You change what lens? You can change the power of a magnification of the ocular objective lens, not the ocular lens, because the ocular lens has a fixed A value. It has a fixed number, like A10, for example. But you can change many times. The power of the objective lens. You can use this one and this one and this one. And if you are in the lab and you want to start study any object or any cell or any tissue, you will start using the power, the smallest power first. Then you move to the strongest power, then to the strongest one. You move to stronger one, then to strongest one. Yani if you are studying the blood, for example, you will use the lens number 1, which has 20x. Then you see the blood, a magnification will be what? If you use the objective lens with 20x and the ocular lens with 10x, what is the magnification power? Who can calculate? If you use the objective lens with 20x, power of magnification 20, and ocular lens with power of magnification 10. So the total magnification of the microscope will be what? Can you calculate? It is 10x multiply what? Multiply 20x. So it will be how much? It will be how much? It will be, of course, 200. It will be a 200x. The total magnification power will be 200x. The total magnification power of the microscope will be a 200x. So if you use the objective lens with 20x and the ocular lens with 10x, the total magnification of the microscope will be 200 times. You can magnify the object 200 times. Type. If we use the second lens with 40x, if this one will be 40, so the magnification power of the microscope will be what? Will be what? 400x. The microscope magnification will be 400 times. And if you use the lens with 100x, okay, the magnification will be what? It will be what? The magnification power will be a 1000. The magnification power of the microscope will be 1000. Mazen, welcome. You are too late, Mazen. We record. You can see the recording after the session. Okay, we are recording. You can see the recording after the session. Okay, Mazen. Now we are talking, Mazen, about the magnification power of the microscope. We have here a light microscope, and we study its parts. It has many parts. It has mechanical parts like the base and the mechanical part, and also the arm is a mechanical part, and the stage is a mechanical part, and the tube also mechanical part. And we have light parts. The light parts like the ocular lens or the eye lens, okay? And also the objective lens, which has many lenses, not one lens only, maybe 3 lenses or 4 lenses, because every lens has its own magnification power. You can use different lenses according to the power of magnification that you want. If you want to study a slide for a drop of blood, for example, if you want to study the drop of blood, you can start using the magnification power by the objective lens of 20x. And the total magnification of the microscope will be the multiply of the power of objective lens multiply the power of ocular lens. So it will be 20 by 10. It will be 200x. يعني magnification, total magnification of microscope will be 200. And if you use the magnification, the objective lens with the power of magnification 100, if you use this objective lens, the total power of magnification will be the power of magnification of the microscope will be multiply the power of magnification of the ocular lens, multiply the power of magnification of the objective lens. It will be what? It will be 100 multiply 10. It will be 1,000 times. So you can magnify the slide, the sample of the blood 1,000 times. Is it okay? Okay. This is the light microscope. This is the light microscope that we depend on the light. We depend on the light. You can use this mirror. You can use this mirror to collect the sunlight and reflect it here on the slide. Okay. And also, some light microscope work with electricity. Some light microscope work with electricity. You can replace this with a lamp and connect it to electricity. You connect it to electricity. And here there is a lamp, not a mirror. There is a lamp, and the lamp can light the microscope. Okay, there is a lamp here which can light the slide which you study. So this is a light microscope but using artificial light, but the other one is a light microscope which is using the natural light. So we use light, we depend on the light in using the light microscope. So it has this name, the light microscope. Now we can move to another type of microscope, which is the electronic microscope. The electronic microscope. Of course, you can see here the microorganisms are very small, so you need a microscope to magnify them. Of course, you need a microscope to magnify them and to study them clearly. I have to talk here now about the electronic microscope. This is the light microscope, as you see, which we can we can see this light microscope at the school, in the lab of the school, and also you can buy one for yourself. You can buy one from some shops, sell this microscope. Okay, test yourself. Before testing yourself, we can read this note. Yusuf, can you read this note?
Okay. If the magnifying power of the ocular lens
to read the note first. I need to read the note first. You know, when we use the light microscope, you can add a dye or a color or an ink to the sample or to the specimen to color it and to watch it clearly. You know, we can add a drop of ink or a drop of dye to color the sample or the specimen which we study. And this is from the disadvantages of the microscope. Okay, so it is preferred to add dyes on it. It is not preferred to add dyes to examine specimen of a unicellular or prokaryotic organisms. It is not preferred to use a dye because you will kill the living organisms like amoeba, paramecium, and also the fungi. You shouldn't use dye to color it because it will kill the living organs, the prokaryote or the unicellular organ. Now you can read the question, ya Yusuf. Now you can read the question. Not ready, Yusuf? Omar. Can you read the question?
Yes.
Read,
If the magnifying power of the ocular lens is 20x and that of the object lens is 100x, the image that is formed by this microscope is.
if the magnification power of ocular lens 20 and that the objective lens was 100x, the image that is formed by this microscope is a clear, accurate, unclear, not seen. تختار ايه؟ تختار ايه؟ فكر شويه. انت هتعمل ماجنيفيكيشن كام؟ التوتال ماجنيفيكيشن كام؟
100 multiplied 20.
100 multiply 20 يعني كام؟ يعني 2000. يعني كام؟ 2000.
Okay.
So the objective, the specimen or the unicellular organism that you will study, it will be very magnified 2000 times. So it will be accurate. It will be accurate. Is it okay?
Okay.
Because you magnify it how many times? 2000 times. Okay? You magnify it how many times? 2000 times. So it will be very accurate. You magnify it very, very big number of magnification. Now, Yusuf, can you read the second question? And Neema, can you read it?
To examine the types of
sorry, master.
ايوه اتفضل.
Read.
اتفضل يا يوسف. معلش يا نعمه، خلي يوسف هو اللي اتفضل يا يوسف.
To examine the types of white blood cells by the light microscope, it is preferred that its magnifying power is C 10 multiply 100.
طب تعالى نرجع معلش للحته دي شويه. المعاصر
أوكي.
قايل فيها شويه حاجات، هنعيد الكلام فيها شويه.
اوكي.
بيقول لي لو عملت ماجنيفيكيشن بيبقى نوت اكيوريت او بيبقى نوت كلير امتى ويبقى كلير امتى. تعالى نبص على الحته دي كده. تعالى الحته دي المعاصر مزود فيها شويه بس خلينا نقرا. بيقول لي الماجنيفيكيشن باور باور اوف اوبجكت اب تو 1500. لو زادت عن كده هتبقى انكلير. خلي بالك بقى الحته دي المعاصر مزودها شويه عن كتاب المدرسه بس هنقولها. يبقى لو زادت عن 1500 هتبقى الايمج انكلير. وبالتالي السؤال بتاعنا هنا اللي هو الاولاني ده هتبقى الانسر بتاعته انكلير. خلي بالك معايا هنا هتبقى انكلير لان انت عندك اكتر من 1500. ده انت هيبقى عندك 2000 معايا. معلش هنا وعديت ال 1500 الايمج بتاعتك هتبقى انكلير. خلاص
تمام.
طب تعالى بقى نرجع نكمل كلامنا في المعاصر لانه مزود الحته دي والحته دي ظريفه برضه. بيقول لي ايه بقى؟ بيقول لي if you magnify it more than 1500 the image will be unclear because the image become unclear or blurred. طيب this magnification power depend on the magnification power of the ocular and the objective lens. يعني بتعمل مالتبلاي للاثنين الاوكيولار والايه والاوكيولار لينس والاوبجكتيف لينس. واللون بتاعك اللون اللي انت بتحل بيه ايه؟ اللون اللي احنا هنشتغل عليه اهو. The magnifying power of the microscope will be magnifying power of ocular lens multiply the magnifying power of the objective lens. تمام كده؟ دي الحته اللي المعاصر قايلها. طيب بيقول لي كمان if you know that the magnifying power of an objective lens equal 50x and the magnifying power of the ocular lens 10x, calculate the magnifying power of the microscope. تقدر تقول لنا ايه يا يوسف؟ تقدر تقول لنا الماجنيفاينج باور اوف ذا مايكروسكوب تبقى كام؟
هعمل 10 multiply 50.
ايوه تمام. اوكي يوسف نو بروبلم. 10 ملتبلاي ايه؟
50 هتبقى ايكوال 500.
500. طيب الصوره بقى الايمج هتبقى كلير ولا ان كلير؟ اقل من 1500
كلير.
تبقى كلير كده تمام. less than 1500 تبقى كلير. تمام تمام. طيب تعالى بقى نروح تاني للاسئله.
اوكي.
طبعا بيقول لي methods to obtain a clear image specimen under the lenses of the light microscope. ليه لو حبيت تعمل الصوره اوضح تعمل ايه يعني؟ Scientists found that the best method to examine the specimen more clearly in the by increasing of the contrast between different parts of the specimen by عايز ازود الكونتراست اعمل ايه؟ changing the level of light. اللايت intensity ممكن اغير اللايت intensity وممكن اغير الديستنس. احرك الايه؟ الادجست احرك الادجست up and down until I have او using dyes. I can use also dyes. استخدم صبغه حاجه تلون العينه. The dyes are used to stain the specimen and become more clear. بس ده مش هينفع مع living organisms زي الاميبا مثلا والباراميسيا او مثلا البكتيريا او مثلا الفنجاي عشان هتعمل كيلينج للليفنج اورجانيزم. فيفضل لما تكون بتعمل examining living organism don't use the dye or don't use the ink to color the specimen.
اوكي.
طيب تعالى نروح
اكزام.
الاكزام. لو قلنا هنا الانسر انت قلت كام؟ اختيار الاحسن ايه؟
سي
اللي هو هيبقى 1000 مش كده؟ عشان ما يعديش ال 1500 عشان ما تعديش ال 1000 500 ولكن ده اقوى حاجه عندي اقدر اوصل لها. هتبقى 1000 تايم. تمام. طيب تعالوا نروح للالكترونيك مايكروسكوب. الكترونيك مايكروسكوب طبعا هنا steps to study the membrane in the onion, the epidermis membrane from the onion. في غشاء شفاف كده اسمه epidermis membrane موجود between the leaves of the onion. يعني ورقه البصله كل ورقه ورا الثانيه في فاصل بينهم بغشاء شفاف كده اسمه epidermis membrane. ده اسهل حاجه ممكن اطلعها واعمل لها ستادي بالميكروسكوب. لو انت عندك في اللاب بتاع المدرسه او في اللاب لو انت عندك في البيت ميكروسكوب من اجمل التيشوز اللي ممكن تعمل لها ستادي واضحه جدا التيشو بتاع الابيديرمس بتاع الانر ليفز اوف ذا ذا اونيون. تقدر تاخد الغشاء الشفاف دوت وتاخد منه جزء صغير باي فورسبس وتحطه على سلايد وبعدين تبدا تحطه على الميكروسكوب وتعمل له دايم وتحط له دروب اوف انك عشان تشوفه بوضوح اندر ذا لايت ميكروسكوب. هتشوف طبعا شكل التيشو بهذا الشكل. هتشوف الشكل دوت عامل زي خلايا النحل كده او عامل زي الحيطه زي البريكس بتاعت ذا وول بريكس. هتلاحظ ان في حاجه غامقه كده في كل خليه. every cell has a dark body. What is the name of this dark body? Can you think? What's the name of this dark body? Can you predict what is the dark object or the dark sphere here in the middle of every cell? The dark sphere in the middle of every cell is the nucleus of the cell. The nucleus of the cell, which is the most clear object or the most clear organelle in the cell under the microscope. طيب تعالوا نروح للايه؟ يقول لي the opposite figure illustrates the method to examine a specimen of plant tissue زي الأونيون مثلا. by the light microscope after adding a drop of iodine. طبعا انا هعمل coloring بالايودين to this specimen. And then it's covered with a cover slip with a certain angle as shown in figure. ماشي. يعني انا هحط العينه التيشو بلانت تيشو واحط عليه الكفر دوت. الكفر سليب عباره عن بيس اوف جلاس كده شفاف غشاء رقيق من الجلاس. طيب الاختيارات بيقول لي ايه بقى؟ by cover slip by a certain angle as shown in figure 2A. انا بحط الكفر سليب بالانجل دي ليه؟ to see the specimen with original size. اكيد اعتقد انها ايه؟ لا امال انا بستخدم ميكروسكوب ليه؟ انا بعمل اوريجينال سايز؟ لا طبعا. طيب to reduce the presence of air bubble دي ممكن عشان بحطها بانجل دي تنفع. دي تنفع عشان كده بحط السبيسمن بانجل معينه دي تنفع اجابه تكون اجابه صحيحه. نشوف الباقي كده. to reduce the size of the specimen طبعا لا اكيد لا مش هقلل حجمها ده انا عايز اكبر حجمها. to more clear and transparent برضه لا يبقى الاجابه الصحيحه to reduce the presence of air bubble يبقى احطها بانجل كده عشان ما اطلعش air bubbles جوه ايه السبيسمن او العينه اللي انا بدرسها. يلا يا نعمه read the second question.
لسه معلش مش المفروض تكون دي to be
ايه؟
clear and مش المفروض to be clear and transparent.
لا هو عايزك تقول له ليه بيحطها بانجل؟ ليه بتحط الكفر سليب ده بانجل بتنزلها بالراحه كده بانجل؟
اوكي.
ماشي؟ فانت بتمنع تكون فقاقيع هواء وانت بتحطها بانجل كده. عارف انت عملت الموضوع ده قبل كده لما كنت بتحط الكفر او السكرينه بتاعت الموبايل. حطيت سكرينه بتاعت الموبايل قبل كده؟
ايوه
ركبت الموبايل سكرينه قبل كده؟
ايوه
شفت الراجل وهو بيركبها لك بيحطها بانجل كده معينه
ايوه
عشان ما يتكونش ايه؟ اير بابلز. صح؟
صح صح ايوه.
يبقى هنا الاجابه ايه؟ to reduce the presence of air bubbles. تمام؟ زي نفس الفكره بتاعت السكرينه بتاعت الموبايل. يلا يا نعمه can you read the next question? Are you ready نعمه؟
The relation between the number of cells that are impaired by the light microscope and the magnifying power of the used lens is
خلي بالك من الكلام نقراه ونفهمه كويس. The relation between number of cells يعني انا ممكن استخدم big number of cells وممكن استخدم small number of cells صح؟ يعني ممكن في السبيسمن بتاعتي في العينه بتاعتي يكون فيها 1 سيل اونلي وممكن فيها 100 سيل. ماشي كده؟ طيب. The relation between the number of cells appeared by the light microscope and the magnifying power. كل ما ازود الباور العدد بتاع السيلز اللي هشوفها اكتر ولا اقل؟ فكري معايا كده. انا مثلا هستخدم باور مثلا 20. ثانيه واحده انا هستخدم باور 20 مثلا في الاوبجكتيف لينز. لقيت عندي مثلا ايه 50 سيلز 50 سيلز او 60 سيلز. طب لو استخدمت باور 40 النمبر اوف سيلز اللي هشوفها اكتر ولا اقل؟ اقل. تمام. يعني مثلا هشوف 20 سيلز بس او 10 سيلز بس. طب لو استخدمت 100 اكس ده انا ممكن كده اشوف 1 سيل اونلي لان انا بقرب جدا وبشوف عينه اقل ايه عددا في السيلز. يبقى الريليشن دي دايركت ولا انفيرس؟ انفيرس ريليشن. يبقى الاجابه انفيرس ريليشن اجابه صحيحه. الاجابه انفيرس ريليشن اجابه ايه؟ صحيحه. شكرا يا نعم. انفيرس ريليشن يعني ايه الكلام ده بقى يا يوسف؟ يعني as I increase the power of the objective lens او the magnification power of the microscope as the number of cells that I see decreases. The number of cells decreases. لكن لما استخدم اللور باور هشوف عدد سيلز اكتر. لكن لما استخدم السترونجر باور هشوف عدد سيلز اقل لكن هشوفها اقرب. تمام؟ طيب الكترونيك ميكروسكوب بقى بيعتمد على ايه؟ لا ده بيعتمد على حاجه ثانيه خالص. لان الباور بتاعته بقى مش 1000 ولا الكلام فاضي ده ولا 1500، لا ده بيوصل 1 مليون تايمز. وده هتلاقيه في الجامعات الكبيره فقط، مش اي جامعه كمان. في الجامعات الكبيره فقط ممكن تلاقي فيها الكترونيك ميكروسكوب. تمام؟ فبيقول لي الالكترونيك ميكروسكوب يعني تم تصنيعه اول مره سنه 1950، يعني في القرن العشرين، في منتصف القرن العشرين 1950، يعني بقى له 70 سنه فقط تقريبا. The scientists started to study the make and use the electronic microscope. طب الفكره بتاعت اللايت ميكروسكوب كانت ايه؟ كانت اللايت depend on the light. قال لك لا انا هنا depend on the speed of electrons. بدل اللايت انا depend on the the electrons electronic beam. Depend on using a beam of high speed of electrons instead of light. خلاص مش هستخدم اللايت. انا هستخدم ايه؟ I will use as high speed electrons. شعاع من الالكترونات. بدل اللايت هستخدم ايه؟ an arrow او a speed of electrons. high speed of electrons. وبالتالي هيبقى اكثر دقه جدا. type of lenses بقى كان هناك عندي عادي جلاس لينس، كونفكس لينسز، لكن انا هنا هستخدم بقى حاجه ثانيه خالص اللي هي الكترومagnetic لينس. electromagnetic lenses that control the electric beam of the electrons. اوكي يبقى انا هستخدم نوع من اللينس اسمها electromagnetic lenses. طب الفانكشن بتاعه ايه؟ الفانكشن بتاعه clarifying the cellular components that had not seen before. ده انا هكبر بقول لك 1 مليون تايم. انا اقدر اعمل 1 مليون ماجنيفيكيشن باور. الماجنيفيكيشن باور كان ريتش 1 مليون تايم. وبالتالي اقدر اشوف حاجات عمري ما كنت شفتها قبل كده. I can see the very small organelles inside the cell نفسها. ده انا اقدر ادخل جوه السيل نفسها واشوف ايه اللي جوه السيل. Knowing more accurate details about the cellular structure had been known before that had been unknown before. حاجات احنا بنشوفها لاول مره بالالكترونيك مايكروسكوب. طب الالكترونيك مايكروسكوب ده نوع واحد ولا اكتر من نوع؟ في اكتر من نوع. طبعا الماجنيفاينج باور بتاعته بتوصل 1 مليون تايمز 1 مليون تايمز or more from the real size. والبروبرتيز اوف ذا ايمج، الايمج مش عاديه بقى، لا ده الايمج بقى بتاعتك هتبقى received on a highly contrast image and comparatively image that produced by the more than the light microscope. يبقى the magnification is bigger، بيوصل 1 مليون times. there is highly contrasted and comparatively to the light microscope. يعني الصورة أكثر وضوحا وأكثر تكبيرا. طبعا due to the shortness of the wavelengths of the electronic the electrons. The beam of electrons have short wavelengths, so shorter than of course the normal light. وبالتالي بشوف حاجات أكثر دقة وأكثر وضوح. Compared to the light, the normal light ray, and it can be received on a fluorescent screen. اقدر استقبل الصوره on a screen زي التي في كده. or highly sensitive photographing board، ممكن استقبلها على لوحه استقبال فوتوغرافيه. يعني سكرينه برضو بس فوتوغرافيه، يبقى ممكن استقبله على fluorescent screen زي التي في كده او highly sensitive photographic board. طب النوعين بقى بتوع الالكترونيك مايكروسكوب، في نوع اسمه سكاننج الكترونيك مايكروسكوب، وده بيعمل مسح للاوتر سيرفيس اوف ذا سيل. بيعمل سكاننج للاوتر سيرفيس ذا سيل، ذا سيل سيرفيس فروم اوتسايد. لكن الاقوى منه بقى الترانسميشن الكترونيك مايكروسكوب، ده بيعمل ستادينج ذا انترنال ستراكشر اوف ذا سيل. يخش جوه السيل ويعمل لها ستادي، وبالتالي اكثر قوه بكتير. فبيديني هنا بيكتشر كده، فوتو بتعبر عن مثلا دروب اوف بلاد by the scanning microscope وفيها white blood cell زي ما انت شايف الوايت بلاد سيل اهي. وهنا لما دخلت جواها بقى وعملت سكاننج بالترانسميشن مايكروسكوب شفت السيل اكثر وضوحا، دي الديتيلز الدقيقه جدا. يبقى واحد سكاننج بيسموه الناسح، وواحد ترانسميشن بيسموه المخترق او النافذ. ترانسميشن هنا معناها المخترق او النافذ. طيب the scanning microscope micrograph of the white blood here blood cell using the scanning electronic microscope the magnification power 3500x. يبقى انا عامل magnification لوايت بلاد سيل 3500 times. لكن هنا بالميكروسكوب بقى الترانسميشن ميكروسكوب the white blood cells using the transmission electronic microscope the magnification power هنا كام بقى 8900x، بكبرها تقريبا 8900 times، وبالتالي الوضوح هيكون اكثر. ده انا جايب الديتيلز اللي جوه الوايت بلاد سيل، وبالتالي more details I can see more details from the previous figure. We can notice that the micrograph of the white blood cell is more clear by using the transmission electronic microscope due to the easiness of distinguishing its internal components. We can see the internal components very easy. So the transmission electronic microscope is more stronger and more accurate than the scanning electronic microscope. Here we have a compare between light microscope and the electronic microscope. In the light microscope, we depend on the light, sunlight. In the electronic microscope, we depend on a beam of high-speed electrons. Okay, and the type of used lenses, we use glass lenses, convex lenses, but in the electronic microscope, we use electromagnetic lenses. And magnification power reaches 1500 times in the light microscope، واكتر من كده هتبقى not clear. لكن الماجنيفيكيشن باور هنا reaches 1 million times. الويف لينس هنا لونج ويف لينس، اللايت العادي لونج ويف لينس، لكن عندي في الالكترونيك مايكروسكوب فيري شورت ويف لينس. طبعا الالكترونيك مايكروسكوب اقوى بكتير. Observation of the specimen we can't see through the ocular lens، لكن we can see the specimen here on a fluorescent screen or highly sensitive photographic board. الباور اوف كونتراست هنا لو كونتراست باور، وهنا فيري هاي كونتراست باور. طبعا الفانكشن magnifying microorganisms, non-living organisms, examining large size cells and tissues, mix slides and examine it, but not more than 1500 times. لكن هنا I can see more clearly define the cellular components which cannot be seen by the light microscope, and to know more accurate details about the structure of the cell and which we don't know before. And here we come to the end of the session يا شباب. The session will end now. I want you to watch the recording and to solve some questions on the microscope. We'll make some exercise next session إن شاء الله. السلام عليكم ورحمة الله.
السلام.
وعليكم السلام.
Try to solve some exercise.